Hello and welcome everyone to our today's lecture on parameter inference. Today we will talk about Bayesian inference for biological models, and we will go into how to do that for commonly used SPML models, but in using the Python tool called Bioscrape to run Bayesian inference so that you can identify the parameter values of your model from the experimental data. Overall, this lecture will not be about going into the details of Bayesian inference or the particular method called Markup chain Monte Carlo sampling that is used commonly for Bayesian inference. So if you're interested in the theory, the details of the theory for how this works, I can refer you to some material, but this lecture will mostly focus on the basics of Bayesian inference, why it makes sense overall, and how we can use it, and how we can use it in the Python tool called Bioscrape, and how it implements it, and then we will move towards actually demonstrating the tools for some data and identifying parameter distributions from that data. So the parameter inference problem can be looked at in this cartoon figure here, where if you have a biological process in the center here, and you create a mathematical model that's an abstraction of the biological process, and that abstraction is what you are interested in modeling from that biological process. And now what you can do very fast and very easily is that you can run the mathematical model in your computer. So that's analysis, and also compute different kinds of properties from the model. You can also do prediction. So if you have a fairly credible model, you can run your model and predict what you might see in an experiment. And all of these you can run in your computer. So you can run using various software tools. You can do various kinds of things with your mathematical models. So this analysis and prediction part is very fast. However, to validate the models, to validate this mathematical model, we need to learn the physical reality, and that's the biological process. And that's why we can use the experimental data that's coming out when we are actually doing the experiment for this biological process, and we want to learn or infer the parameter values of our mathematical model from that experiment. And that is this arrow at the bottom, this cycle. And the issue here, and as you will see today, is that this is not a fast process like just running or simulating your model is, because this involves simulating your model thousands of times and running other computations. So this will be the focus of today's lecture, that we want to infer the parameters given the data, and we want to feed those parameters back to our model that we were simulating, so that now we can go through this pipeline of analyzing and predicting with much more confidence, because now the parameters of that model would be trained or would be set according to the reality of the data that you just observed. What are some of the common ways in which parameter identification is done in the literature? The first one is on the very left, nonlinear optimization, where what you would do is you would run an optimization algorithm that would find optimum values for each parameter in your model that would satisfy the data. And as you can imagine, this would get computationally very expensive very quickly, because we have nonlinear models for biological systems, and those can get huge, and analytical solutions don't exist for nonlinear optimization of these models in general. And so this can get computationally very expensive. A very common approach for parameter identification or model fitting is our machine learning methods. And what they do there is you would, for example, train a neural network model that would have maybe thousands of parameters in that model that would regenerate or capture the experimental data. But there are a couple limitations there as well. You need lots of data, number one. Number two is that the model that you create is not as interpretable anymore, because that model has weights, and those weights and different parameters in the neural network model, you might not be able to connect those back to the mechanistic description of your biological system. And so going through that cycle of design, build, test is even harder, because once you identify the model, you want to run the simulations or get some guideline towards the design of the system as well. And for that, you need to connect those values back to the mechanistic reality, and you need to know what parameters you identified. So that leaves us with the third one, and that's what we will be talking about today, and which is Bayesian inference. Bayesian inference is when, instead of finding out a unique value for the parameters like we were trying to do in nonlinear optimization, you instead focus on determining the distribution of parameter values that could be possible. So in this problem, we deal with everything probabilistically. So we say that there might be a probability distribution for each parameter given the experimental data. And that distribution is what we need to find out. So we will talk about this because this can be used for the chemical reaction network models that we can easily build for biological systems. And it can be used then to identify those parameters from those models and then connect back easily into the design, build, test cycle. How I like to learn about Bayesian inference is, first you think about sampling from a probability distribution for a second. And I will describe the problem and we'll leave it with you for a few seconds, and we'll give the answer in the next couple slides. So the problem is, how do you sample from a known probability distribution? So let's say you have a multi-dimensional Gaussian probability density, and you know this formula is analytically known. So you know that the probability is proportional to this, where x could be a vector. So it's like a multi-dimensional probability density. Now, the goal here is that we want, if I say, give me 1,000 samples that are distributed in this manner, that have the probability distribution that looks like, or that follows the Gaussian probability density. How can you implement such a thing in a computer simulation is the question. The counterintuitive thing is it's not as easy as it might sound, because although you have this expression, you have everything you might need, but how do you implement this so that you have a function where you can ask for 100 samples or give me 1,000 samples, something like that. So think about something, and I will share the answer in the next couple of slides. Okay, so One way to do it is what's called Monte Carlo sampler in this literature, and which you might have guessed is random sampling. So what you do here is that you keep on randomly sampling, or in other words, you keep on drawing independent and identically distributed samples each time, and then check whether it falls under this probability density and accept or reject each sample. You keep on doing this for a large number of samples, and after a large number of samples, you would have the required number of accepted samples. When you have that, you can stop sampling, and that's it. And that's called the Monte Carlo sampler, where you're just randomly sampling everything. And at the end, you have a representative sample for this Gaussian probability density. So, of course, as you can imagine here, it's not a very efficient way to do it, because the next sample that you're choosing is always random, irrespective of what happened in the previous sample, right? And that's what MCMC does smartly, is that every next sample that the Markov chain Monte Carlo sampler that we will be using proposes is dependent on the previous sample as well. I will not be going into the exact formulation of how it's dependent on that, but we will look at how the overall Bayesian inference works and how it decides whether to accept or reject a sample, and how that affects your next sample. And we will be focusing more towards examples of running this. And as I said earlier, it's a very interesting theory. So if you're interested in learning more about it, I can suggest material that you can go through. But with the overall idea in the mind, that Markov chain Monte Carlo is a sampling technique that does sampling of probability distributions smartly or efficiently. We can again look at the parameter inference problem that we were looking at earlier. And now we can say that the parameter inference problem is actually finding out the probability distributions for the model parameters given the data. So instead of saying that you want to find out exact model parameter values given the data, what I'm saying now is that our goal is to find out the probability distribution for the model parameters given the experimental data. And that's what we can formulate using Bayes' rule. On the left-hand side is the thing that we want to compute. That's probability of theta given the data. So theta could be the set of parameters that you want to identify. So clearly, the left-hand side is our goal. We want to find out that probability distribution. And what do I mean by we want to find out? Computationally, that just means we want some representative number of samples from that probability distribution. And we will see then how the sampling problem comes into play here. So we want to find out this. Using Bayes' rule, we know that we can write this probability distribution as this on the right, where now let's see what each term here is. So the first one is probability of data given the parameter value. So although the left-hand side probability distribution is not so obvious how to compute, right? That's the whole problem. But probability of the data given the parameter value seems something that we can compute. How? Because the parameter value is, it says it's given, so you can fix a parameter value, and then you can compute what's the probability that you see this data from your model. So this is called the likelihood probability distribution. The second term here is just the probability of data. So what's the probability that I see this parameter set and that's called the prior probability distribution. So because this doesn't depend on data and is known prior to running this parameter inference, we give probability distributions to each parameter. So each parameter can have some probability of attaining some value, and that's the prior probability distribution. And divided by the generative probability, that is the probability of the data. So that's what's the probability of this data set being true. Again, on the left-hand side, this is called probability of parameter given the data. So that's the posterior probability distribution. That's what we want. And then we have it in terms of something that we can compute, the likelihood and the priors that we know. Expanding that further, just looking at the numerator, because we don't want, of course, if we can have it, it's great, but we don't really want an exact analytical description of the probability distribution. We only want a representative sample from that probability distribution. So if we can write that it's just proportional to the product of the likelihood and the prior, that's the first line, and taking log on both sides, what we get is that the log of the posterior probability distribution is proportional to the log of the prior plus the log of the likelihood. And this is exactly what we will be using. And this is exactly what Bioscrape also computes. So Bioscrape would compute for you the log prior, the log of likelihood, add those together, compute the log posterior for each parameter value, and then run this MCMC so as to converge to distribution of parameters that could be representative of your of your data that you started with. With this in mind, and now looking back again to our original problem, parameter inference, we can formulate the parameter inference problem in the following manner. We can say that the parameter inference problem is to generate M samples from the posterior probability distribution. And as I said earlier, we don't really need to find out exactly the analytical description of that probability density function. We just need M samples that are representative of that density. So that would be how we can solve the parameter inference problem. You just need to get samples for the posterior probability density. That's the probability of parameter given the data. And we know from Bayes' rule that that is actually just proportional to the likelihood and log likelihood summed with log prior. So with that in mind, we can implement this sampling algorithm with the MCMC sampler, which I didn't really talk about, which is interesting theoretically, but it's just an efficient way to sample from a distribution, from any distribution. But here, we want to sample from the posterior distribution. When I mentioned earlier that MCMC does it smartly, what it does is it uses a cost function that is defined. So the cost uses the values of those cost function as a way to navigate the parameter space. And this cost function is usually dependent on how you define it. For the simple deterministic inference examples that we will be talking about, the cost function today, for most of the cases, is just the norm of the difference between the data and the simulation. So for each parameter value, the MCMC sampler would compute this norm of the difference between the current simulation and the data. That will give some value to the sampler. And then it will use that value to propose the next sample and will try to minimize that cost. And once it reaches certain minimum level of the cost, it will keep trying to sample and then at the end give you this representative sample of the parameter distribution that the posterior, that's from the posterior probability density. So let's jump into how this might look when we implement it. Every MCMC sampler that we implement for our parameters can be thought of as a random walk, although it's not really random as we saw, but it can be thought of as walker, which walks around in the parameter space. So what I have here is, on the left, is some parameter that I want to estimate, right? So I start with some initial guess, which is somewhere around 0.8 here. And then what it does is this is just one walker. So this is exactly what's happening inside MCMC. So it proposes a certain value, and it keeps on proposing new samples. You see, it's trying to explore all of this space. And why it's not going here, for example, on the top? So it suggested some parameter value that gave a very high cost. So it came back down in the next sample, and then it didn't really explore that area anymore. So it's exploring areas which give lower cost. So this is just one walker. So that's just one chain being formed, one Markov chain being formed that is exploring the parameter space. And you can see exactly what's happening here. It's exploring the parameter space. And after certain number of steps, you might see that this converges. So it doesn't really try to explore anymore, diverge anymore, and it converges to a smaller region. And once that happens, we can say that now we are able to get a representative sample from the posterior density function, and so we can stop the sampling algorithm. So of course, the higher the number of walkers, you are increasing your chances of finding the actual posterior density, because higher the number of walkers means instead of one walker starting here, you start with 10, 100. So all of those 100 in parallel explore your parameter space. And if all of those 100 end up at the same place, you can be fairly confident. But it always, of course, depends on the data, how complex the data is. But ideally, you would want higher number of walkers. The trade-off is, of course, the computational cost that comes with it. Number of steps is easy, is for how long you run that chain. So these are some parameters that you can tune while running your parameter inference. So for example, on the right-hand side, I have same parameter inference, but with 10 walkers. So here you see it's dark because now 10 walkers are starting at the same place and starts at a value around 0.7. And all of them start at exactly the same place, but very quickly, within 10 or 20 steps, it's already explored most of the space you see here. It's not going beyond these values because those are outliers, and it's just exploring this area here. So it started with something like 0.72 or something, and then it converges to something else, 0.75 or something like that, whatever. But it can converge at different places depending on the experimental data that was given. But the chart here is trying to tell you that there's two parameters, the number of walkers and the number of steps in the chain, are important to tune depending on the computational cost and the accuracy that you want. There are other parameters for judging the accuracy of the sample, and those are statistical measures that you can run to find out whether you can call this chain long enough, how to know when you actually have a representative sample or not. Those are also computed by Bioscrape inference that we'll talk about, but I won't go into the theory of judging when the convergence has been achieved. But that's, again, another interesting problem. So finally, an overview of how everything functions is we load the experimental data using Python pandas package. Then you store it as data frames or create a list of data frames if you have multiple conditions. Then Bioscrape inference uses the internal Bioscrape simulators to compute the log likelihoods and run all of the other computation. It also interfaces with the Python MC package. That's the MCMC sampler. So we need that intelligent sampler That comes from this Python MC package. And then using that, it runs the whole Bayesian inference algorithm to give you out parameter distributions that you can then plot and visualize with this corner package. So it's a culmination of all these packages together let you do parameter inference. With that, I will point you to the installation steps. So you need MC as mandatory. The others are plotting. And you also need pandas to load your experimental data. For Bioscrape, even if you have it installed already, or if you don't have it installed, just go to the GitHub biocircuits/bioscrape GitHub and clone the repository over there and run python setup.py install to install the software package. Then you'll be ready to run all the Jupyter notebooks that we will be using for the demonstration next. Yeah, that's an interesting problem of, I would say, research that people are interested in, because as the data gets bigger, the computational costs are huge because you're running the simulations thousands of times. So if you're trying to estimate 5 parameters versus when you're trying to estimate 500 parameters, the computational cost is huge there. So there are parallelizations that are possible, especially with Python MC. It's built natively to be parallelized, and so if you can take advantage of that, and any other computationally like where you can increase your computation speed is always helpful. Yeah, again, that's a good question. And when you don't have any, so prior, as the word itself says, like if you don't have any information at all, you can just start with uniform prior. And I didn't go into the details, but I'll talk about it in the workshop. But uniform prior is something where you just specify the maximum and the minimum range of your parameters, and then you're basically telling the sampler to only explore in that range, but you're not telling anything more than just those minimum and maximum values. And so if you have information from your, so for example, for your experiment, you know that some values are more likely than others, you could think about what the probability distribution might look like. And it could be a Gaussian distribution, or it could be other commonly used probability density functions. And so if you have that kind of an idea that maybe your parameters, you know, experimentally follows a Gaussian distribution, that is, there are some values near the mean that are more likely, then what you can do is you can use a Gaussian prior for that parameter. But in case when you don't know anything, you can just give a uniform prior, and that's what it is. It's like a random prior where things can be sampled randomly. I would say if you have an intelligent prior, you are helping the algorithm a lot. But if you're not, it doesn't really matter too much. I would say if there is only a few number of parameters, because all this is very fast and you can run thousands of simulations, even like it's like sort of brute force, but you won't notice the difference. But I would say when you have large number of data and big model, choosing intelligent priors might play a role in how quickly you converge. Because without that, like you're not telling the algorithm anything, and it's exploring everywhere. And I would say there are times when you have a multimodal posterior, so it might be possible that there are 2 different values or 2 different regions that the parameter can take values in, and those are separate from each other. And so those are the cases where it gets really tricky to run this parameter inference. And I would imagine that those are the cases where you might need to think about priors more closely. Yeah, again, very good point. And as we've been doing in this class, we've been building chemical reaction network models, which have chemical reactions for all of the different things. It's like it might be a 10 or 20 state or even bigger model, but when you do it experimentally, you might just be getting one output measurement, as William mentioned. And so the problem of identifiability is that you can theoretically, first of all, analyze whether or not your system is identifiable. And what that means is if you can identify the parameter values uniquely from the given measurement. And if you think about it, what might happen a lot of times is that a combination of parameters might change together. So if like you increase one parameter and you decrease the other one, the change in the output that you're looking at there might not be any change. And so that tells us that you cannot uniquely identify both the values, right? Because there are multiple sets of parameters that give you the exact same output. And so how do you distinguish between those sets? And if that's the case, then you say that your model is unidentifiable, and you have to live with it. You either get more data from other outputs, or you try to figure out what the unidentifiable sets are. So what are those lumped parameters that change together, so that you only try to identify those lumped ones. Yeah, and so it's especially a challenge for biological system models where having one or two outputs is very common. And that is why it's very common also to use, I would say, phenomenological models rather than detailed chemical reaction network models to identify model parameters. So like the models that are reduced and you modeling only a couple or 3 species for instead of modeling 10 reactions for transcription translation, you just have 1 or 2 equations modeling directly the protein equation using some Hill functions. So that's why they are common, because they help in getting those parameters, like all of those parameters are then lumped already. And so those become helpful. First, I would say that you can choose the number of parameter, or the what parameter set you want to identify. So for example, you have 10 different parameters and you're fairly confident in the values for 5 of those, and you only want to know what the values are for the other 5. So you can say that those are the ones that you're trying to estimate. And when MCMC is running, it will try to only find out the posterior probability density for those 5. So what that means is for all 5 together, it will change the values individually, of course, and then simulate the model for all of the parameter sets. And then what I mean is change the values of the parameters that you're trying to estimate, run the simulation, compute the costs, and do all of that, and then give you out so the result then would also be distributions for 5 different parameters in that case.